Hello, everybody. My name is Maxine, and I'm from the John Parkinson Lab at the University of Toronto. Today, my story is about my current project, which is identifying efficacious dietary additives to improve poultry gut health. Antimicrobial growth promoters have been used in the poultry industry because they have this overall antibacterial effect. It's been found to decrease production of toxins by intestinal bacteria and reduce intestinal infections, such as those caused by Clostridium perfringens, Campylobacter, or Salmonella. It's also been found to increase feed conversion and bird growth. However, use of these antimicrobial growth promoters are linked to a rise in pathogens developing antimicrobial resistance mechanisms. And so the industry has explored using probiotics as alternatives to antibiotics. There are two main types, single-strain and microbial consortia. Microbial consortia is fed to birds as a collection of bacterial members. My overall aims for this project is to identify alternative additives for poultry production that can exert colonization resistance and improve engraftment in the gut. Ultimately, we can increase our understanding of the microbiome and its impact on host health and immunity. Specifically, we are exploring how probiotics can impact both the composition and function of the microbiome. Here, we conducted animal trials with Dr. Doug Corver from the University of Alberta. We separated chickens into groups and fed them either bacitracin, which is an AGP commonly used in the industry, Avigard, which is a microbial consortia that is commercially available, a probiotic yeast, and NPP1. This NPP1 is still in development. It's based on a Bacillus pavillus strain. Then we harvested samples from the jejunum, ileum, and the ceca at days 10, 24, and 40. And these trials were repeated in the corn and wheat-based diet. Here, this is a taxonomic breakdown of sequences generated for each GI site, treatment, and the individual time points. The first three rows represent the 16S rRNA sequences taken from the jejunum, ileum, and the ceca, whereas the last fourth row represents the transcriptomic sequences taken from the ceca at days 10 and 24. And we see here that lactobacilli in this light green is predominant in the small intestine, in the jejunum and the ileum, while in the ceca, there is an abundance of Ruminococci in dark blue and Lactospiraceae in this light blue from the Firmicute phylum. And this difference from the small intestine to the ceca is likely due to the fact that the ceca is where microbial community is at its richest, and it's where most of fermentation happens. So the short-chain fatty acid producing bacteria, such as the Firmicute phylum, are most abundant at this site. While in the small intestine, mostly digestion of plant matter takes place. And I'd like to point out that within the Avigard samples, or this treatment group, we see Coriobateriaceae in this light pink from the Actinobacteria phylum, and this Veillonelliceae in this lighter purple, which is a gram-negative Firmicute bacteria. And you can see that this is in both of these bacteria is in higher abundance as compared to the other treatment groups. And we do know that adult hens are known as efficient donors of gram-negative Firmicute along with Actinobacteria. And we see again that Avigard samples based on the transcriptomic data set that promotes Cella mondaceae, a gram-negative Firmicute again. And this all suggests that perhaps Avigard is a successful as a microbial consortia in providing baby chickens, a microbial community that can mimic communities from adult chickens. Here in panel A, there are box plots of observed OTUs in samples fed with a corn diet. In other words, observed OTUs just means the number of unique bacterial members detected, grouped by GI site, time point, and treatment in different colors. And we can see here that species richness in the microbiome is largely driven by GI site and bird age. The ceca is much richer than the small intestine, and as birds age, their microbiomes increase in richness. And treatment wasn't found to be a significant factor here in influencing alpha diversity, and these observations were found to be the same among birds fed with the wheat diet. In panel B, these are NMDS plots based on Bray-Curtis distances for all the samples. We see that microbial communities at each GI site develop distinct communities, and that the ceca has a more distinct separation from the small intestine. The next factor that may have an influence on the microbial structure is time point or bird age. However, treatment and diet don't seem to affect community composition structure. Finally, in panel C, these are PCA plots based on microbial gene expression in the ceca, taken at day 10 and 24. And each of these points represent a sample, grouped by treatment, time point, and diet. Here we see that samples treated with Avogard may be expressing genes quite different from the other treatment groups. And here we also see that gene expressed can shift from day 10 to 24. And we do see here that diet does not obviously influence gene expression in the microbiome, similar to what we saw in that diet does not seem to influence microbiome composition. And thank you for listening. Here is the QR code for my LinkedIn, and here is a QR code for the published article that precedes this project and explores in depth how antibiotics impact the bird gut microbiome in terms of composition and function. Thank you.